Good afternoon, everyone, and welcome back to the lesson on epigenetics. So we are actually learning it for nuclear reprogramming, and we discussed about what makes a nuclear program. That is to say, is every cell has an internal program that is usually genetic information. What you mean by it is what genes are expressed, right? And of course, there are also some genes that are completely silenced. Which genes are silenced? And which genes are produced in response to the external signals, right? And what is happening to the genome constitutes the nuclear program. You call it nuclear program because everything is in the nucleus, right? It is about the physical manifestation of information. Genetic information is a cell, and it is the body of a human or any organism. Information manifesting as a physical manifestation of genetic information is the human body or any organism itself. Then, if you talk about one cell, the way the morphology, functions, form, and everything of a cell is the essence of the genetic program. And how is that nuclear program or the genetic program in a way is made? It has to do a lot with epigenetics, right? It's the response to the extrinsic signals is what makes the cell survive and exist, right? If the pH is different, the cell has to respond. If the salt concentration is different, cell has to respond. And there is always this response. And the way genes are repressed, or the genes are activated or remain active, or they are bivalent in the sense they can be repressed or expressed in a cell, is majority of them are dependent on epigenetic modifications. And that is through several of these proteins that we have discussed: histone methylases, demethylases, cystallases, deacetylases, similarly DNA methylases, DNA demethylases, and so on. So what we are, there is, in broadly, if you look at, you can find that the best understood one is DNA methylation, and we have histone modifications, and then nuclear positioning, chromosome architecture as well. In silencing, we also have partly of non-coding RNAs in epigenetic responses. So quickly, we will have a review of imprinting. We already discussed that, I think, at least twice before, and we will discuss that again once. Epigenetic differentiation that depends upon its parent of origin. There is also another definition that comes, which is it's a play with words. The essence is same, right? And we have to remember that these are mechanisms which are not any other changes other than nucleotide sequence change. It could include chemical modification or physical organization and so on. So what happens to imprinting in many of these cells? That is what we are actually talking about. In this case, in normal zygote and IVF zygotes, you will find that imprinting is normal because they are biparental. They're biparental in the sense, you have one set of from father paternal and one set from maternal. What happens to that in SCNT embryos? It is derived from one individual, right? Because it's a somatic cell. But that somatic cell is derived from, without modifications in terms of imprinting, from a male and female. So it does grow to an extent quite normal in SCNT embryos. Imprinting is quite normal in that. There are problems, of course. It's not as it is with the normal zygote, but then you can still think of it. In parthenogenic zygoids, the imprinting is derived from one individual, that's the female, right? And then, therefore, you can have, there will be, if it is derived from one egg, then they both are maternal sets. It's diploid. They all are diploid that we are discussing, and they are both are GG, gynogenetic, and therefore there are imprinting problems. We will discuss why again, you will see. So the problem with imprinting with uniparental stem cells or zygoids is that they may have twice the amount of expression and zero or no expression of some genes, because of which we know that the developmental potential of these uniparental stem cells is not as good. And these zygoids do not give rise to a viable organism. The same is the case with gynogenetic. These may be derived from two individuals. It could be two individuals, but the point is, if you are taking two maternal sets and putting into one cell through pronuclear transfer, then they are gynogenetic, and for the same reasons as in the case, they would also fail in terms of imprinting. They may have overexpression of some genes or underexpression. In androgenetic, they might be derived from two people, but both are paternal, and then that also will result in imprinting problems. So what we will also discuss is nuclear reprogramming is controlled by the imprinting and the other epigenetic mechanisms. So we will have a look at some of these things, and gradually the objective of it is to go through introduction to epigenetic mechanisms. Did I tell you about the course on epigenetic control of epigenetic mechanisms of gene expression or so? The Coursera course. Did I share the link with you? Somebody?
Yes, sir.
Yes. And did anybody try to explore that Coursera course? You can audit the course as well or apply for financial aid. It is a very good source of information. At least once we are halfway through this course, you will be able to follow that course. Otherwise, it may be a little bit complex because it goes from the basic understanding till the level of research. So I hope you will do it sometime in the future, at least, if not now. Otherwise, you can just watch the videos, and they're fantastic presentations and in-depth knowledge. So this is the other kind of other definition for imprinting, you can say. It is epigenetic marks. It is epigenetic mechanisms. That means there could be modifications of DNA, physical or chemical, but not sequence. It's not change in sequence. Okay. And the second one part of the definition is permit monoallelic expression. That means you have two alleles, one belonging to the paternal allele, one to the maternal, and only one of them would be expressed. Either it would be the particular gene could be expressed only from the paternal allele or the maternal allele. Some genes for maternal are maternally expressed, and some genes are paternally expressed like this. So what we are going to do today is we're going to look into these mechanisms, not only about imprinting, but epigenetic mechanisms in general. So genomic imprinting mechanisms, or in general, epigenetic mechanisms, involve two types of modifications. One is covalent, that is DNA methylation, and non-covalent ones could be DNA protein interactions and DNA RNA interactions. Okay. In all these mechanisms, imprinting can happen. We will also have a look at one example, at least in some part today, about DNA RNA interactions. I hope you recollect this picture from the book Watson, and it is about imprinting. And here I'm going to explain what happens. One of it is maternal. A that represent is maternal, and this is paternal. And there are two different genes, not necessarily very close by. They are far away. And the same way is this break. So there are three genes, three parts. One of it is IGF2, that's a gene, H19. And then we have enhancer, and we have an insulator. So don't worry about the actual functions of these genes and so on. We are not concerned about it right now. We are trying to understand the general mechanisms of imprinting or epigenetics in general. So paternal chromosome will also have IGF2 gene, insulator element, H19, and enhancer. There are two genes, H19 and IGF2, right? The insulator sequence, okay, we'll start with enhancer. The enhancer is bound by activator proteins. The activator can actually activate the expression from IGF2 or H19, but never both at the same time. By default, and the activator that is bound to enhancer, the DNA, I hope you remember, it has to loop like this and then activate the transcription from the target genes like this. Okay? If they have to do that, they have to loop, the DNA should loop like this. If the looping is obstructed by any of the protein binding or so, then they cannot loop that far. So somehow the proteins that binds to the insulator can prevent the interaction of the enhancers here with that of the IGF2, right? So the enhancer can typically bind here unless it is obstructed, and obstructed by, say, a protein called CTCF, which would bind to the insulator. So in that case, if CTCF prevents the interaction of activator with the IGF2, then the activators will activate the transcription or gene expression from H19. So in this case, H19 is expressed while IGF2 is not. Let's look at what happens to the paternal chromosome. Enhancer, okay, the insulator region and also including the promoter region of H19 is methylated. As I discussed earlier, most of the proteins are bound or have evolved to bind DNA without any methylation. Of course, there are specialized proteins which bind only to the methylated DNA, but this one is not. So if the insulator region is methylated, then CTCF cannot interact with this region, because of which the enhancer can now interact with the promoter regulatory elements of IGF2 and induce the expression of IGF2. Whereas H19 is repressed for two reasons. Firstly, the enhancer is not interacting with H19, and secondly, the region is also methylated, because of which the minimum expression may also not happen. Okay, so what I want you to take from this is the quantity of expression. If you have a biparental organism, they would have one maternal and one paternal. That means they would have had one X expression of IGF2 and one X expression of H19. If it were, say, gynogenetic one, it would have had two sets, but both are from the maternal one. In that case, maternal allele, IGF2 is off. So there will be 0x of IGF2 expressed, while they would have had 2x that of H19. In an androgenetic one, they would have two sets derived from the paternal alleles. So they would have had twice the amount of IGF2 and 0x of H19. Okay, that is genomic imprinting. Imprinting, that is going to, some genes are expressed only from the maternal allele, some genes are maternal genome set, and some are expressed only from the paternal genome set. So this is something like you have to ensure, it is like it has evolved to ensure that both, only the biparental ones will arise, and there will not be any other possibility. We are going to see several examples of the same, of similar things. So it can be redundant, but it is also important to understand how many are there. So here is what I have discussed, IGF2 and the same example, enhancers, H19, and so on. DMD is, the full form is differentially methylated domain. I wouldn't use the word domain, but otherwise differentially methylated region is good. DMR is a good word to use, because domain is, in biology, we use it for certain structural part of a protein with a function. So differentially methylated means either it could be methylated or it might not be methylated. Okay, the same is what we discussed as insulator. So here are two more examples. I hope you remember we have seen several, we have seen a picture which contains 4 different types of genetic architectures. Where does methylation happen, the genes that are organized, and so on. These are real examples. In B, if you see, there is a set of genome set, you can find it, it is as per the figure, it is given like this, and there is an antisense, there is another gene, an overlapping gene like this. The first one, the top one, codes for IGF2R. R is for receptor. IGF2 is the ligand, and IGF2R is the receptor. And I would also, you also please keep this paternal, maternal, and paternal. So IGF2 is expressed from paternal allele, right? If you see here, IGF2R, that is the receptor for IGF2, is expressed from, IGF2R is expressed from paternal allele. Maternal allele. We'll get to that. Let's finish the expression of this. So there are 2 promoter elements, promoter regions. One of it is for IGF2R, and the other one is for a non-coding RNA called as AIR. All the black ones are supposedly, I think, the repressed ones. So in this case, this region in B, the maternal allele, IGF2R, the regulatory elements of that region is not methylated. Therefore, there will be expression of IGF2R. Whereas the regulatory region of AIR is methylated, so AIR is not expressed. Because AIR is not expressed, SLC22A2 and these regions are also expressed, which we will see. But on the paternal allele, IGF2R is repressed and AIR is expressed. AIR produces, I think it's a non-coding RNA, which further represses these 2 genes. Okay. So what you're seeing is, again, I will just reiterate, IGF2 is expressed from paternal allele and its receptor is expressed from the maternal allele. There are similar examples. This is a NAP115, I think L15 probably. It's a simple one where whether this region is methylated or not. If it is not methylated, NAP is produced, and if it is methylated, it's not expressed. So there are several other examples if you keep seeing it. I'll skip to. But I hope you will want to, we have seen this figure before, and I want you to remember this. During the formation of the embryo, around E is for embryo stage, and 9.5 probably is about the days from fertilization. So if they are doing it in vitro, they know when the fertilization has occurred, they will start counting it as embryo from that day onwards. If they were doing it in vivo, that means a mouse is allowed to mate with another mouse, okay? And they don't know when the fertilization actually occurred. So in that case, they would use days post-coitum. Coitum is for mating. Days after mating. That is how they count. If it is in vitro, they use E, or they might use another one will also come. But I think this is fine. So about around 10 days or so, the first primordial, so zygote has formed. Assume this is the zygote with male and female pronucleus, and they have fused. And then it will start forming an embryo. And somewhere outside or around that, so they will form primordial germ layers, ectoderm, mesoderm, endoderm. And after that, and little more further, they will form primordial germ cells. The zygote that has formed has had parents, male, sperm and egg. This is biparental. And then all the primordial germ layers also are biparental. I mean, the imprinting wise, they have received the same imprints that were made by the parent. And when this embryo is making its own gametes or germplasm, things will change. What happens is all the imprints are erased. That is, demethylation will occur. And if this is a female, because one of the X chromosome is inactivated, that X chromosome is also reactivated. Okay. And what happens is the PGCs occur somewhere here, and they slowly travel in the extraembryonic tissue somewhere outside the embryo. They form, and they slowly move into the newly forming gonads. That is referred to as the genital ridge or gonadal ridge. A genital ridge which will form the gonads. They go in there and they start populating. They will divide multiple times. Okay, this is the stage where they have reached the gonadal ridge and they have populated that. And these are the ones that will serve in the future to form either oocyte or sperm based on the sex of the individual. So during gametogenesis, so the gametes were from here, the imprints were inherited from the parents. Around this stage or so, during this stage, the imprints are erased. Whatever has received, this happens only in the germplasm, right? The rest of the body inherits the imprints from the parents. And during gametogenesis, establishment occurs. Right? During the end of they say initiation, because in sperm it is completed, whereas in the oocyte it is not yet completed. So we will see those things at a later time. But I hope you understood the scheme that happens for imprinting during the early embryogenesis. And the imprints that were made here belong to the person that is making. Right? I hope you remember you saw a picture. I showed you a picture of a pregnant woman. And if this is the woman, and the woman has made a baby, pregnant woman, and the baby has her own germplasm as well. The somatoplasm of the pregnant woman is derived from her parents. The imprint, that is the first generation imprints in this case. The imprints of the baby are made in this individual. And the imprints in the primordial germ cells are assumed there, if they were forming gametes, they are they belong to the fetus. Okay. So how many genes are actually imprinted? This is old data, maybe 100 imprinted genes. Now I think with more research, I think this has been expanded, and majority of them have placenta and involved in placental and fetal growth, and that is why we have seen that maternal alleles, AG and GG have differences. In AG, placenta grows more. In GG, relatively the fetus grows more, and poor placenta. So out of all those things, approximately 30% of them are non-coding RNAs. These non-coding RNAs have several regulatory functions, and that is how the whole developmental processes and several activities are influenced because of imprinted genes. So there are also regions called as imprint control regions, like we have seen LCR, locus control regions. We also saw GCR, global control regions. In this case, when you say imprint control regions, these regions are large scale, which constitute several genes in one location. They are imprinted. These might not be imprinted, but they may be controlled by gene regulation, epigenetic means of control. So epigenetic control and imprint control are almost same, except one difference is here, sex of the parent or the genome set matters. Okay, that is one key difference between how you can see imprint. So here is a website, geneimprint.com. You can go there and try to explore several of these, and you can find with more and more current data. They keep it updated. Some of them are proved and some of them are predicted. Okay, and they show which allele is expressed. So this is how a chromosome 7 is, and these are some of the clusters. Like I said, imprint control regions, you can assume this is a cluster, and so on. And they can be, some of them can be separate also, like these. And the red ones means they are expressed in the from the maternal allele, and blue would imply that they are expressed from the paternal allele. Okay, so we are talking about one chromosome, and one chromosome can have multiple of these loci, and we have several 23, including sex chromosomes. We have 23 pairs. So I hope you can understand that there are multiple genes that are imprinted. Here is a mechanism we discussed in the beginning that we will also have an example about non-coding RNAs and their location or change in the epigenetic control of gene expression. Here is a protein, an RNA, non-coding RNA, ncRNA, which is called as hot air, and it binds to two proteins on the 5' side to PRC2 and the other one LSD. PRC2 is polycomb repressive complex 1 or 2. We have both. And these are just one small examples, okay? So what hot air does is, through its interaction with the DNA, it brings these two proteins to the location, to the target site. And in that target site, these proteins might induce methylation of the histone tails. H3K27 ME3, which I'm unsure about ME3. What does 3 stand for? I'll find out and let you know. But H3 is histone, and K27 lysine at position number 27, and methylation is done. The same way, when the methylation of this locus happens, this particular locus near the HOXD upstream, the gene expression is stopped. The same way, we also have other examples like cyst. We will talk about cyst when we are talking about X chromosome inactivation. It binds to PRC1 and PRC2, and similarly, there is another one called as ANRIL. They can induce the repression of several genes like this. Okay, so the RNA components have two functions. One is to bind to the proteins that can induce histone modifications in this case, but they also have specificity by interacting with the DNA here. And that is how they kind of bring these proteins to the areas where epigenetic control is established. I was talking about genomic imprinting, right? We discussed about IGF2 and IGF-R. And I just want to have a small discussion about it, so that you will also try to be perplexed equally with me. Normal zygote, normal embryo, normal placenta. Androgenetic, big placenta, and embryo is very small. Whereas in gynogenetic one, placenta is small and the embryo is larger. So why are these things happening? And I want to put in one second, please. What I'm saying, try to do is, I want to show you some example, because which will which will find it interesting. IGF2 is a ligand that is secreted, and IGF2 receptor, or the IGF2R gene, that is where the ligand will bind to. So this is IGF is insulin growth factor, I think, and it interacts with IGF2 receptor. So IGF2 receptor from the mom, receptor is on and IGF2 is off. From the paternal side, okay, IGF2 receptor is off and IGF2 is on. Now, if you take AG or GG, you will have twice the amount of IGF2 and 0 amount of IGF2R. In the gynogenetic one, 0 IGF2 and twice the amount of IGF-R. So both these genes are imprinted. So if you delete mother's IGF2R receptor, then the embryo grows big. If you delete only IGF2 gene from the father, then this one grows small. The problem is, the interesting or puzzling thing is, if you delete IGF2 receptor from the mother, right? If you delete both IGF2 and IGF2 receptor, then the offspring size is normal. I mean, there will be a lot of other changes, of course, just based on size. If you see, if you don't have receptor, it is growing huge. If you don't have the IGF2, and growth is less. But if you delete both, you're having normal sized embryo. How is that possible? That is what is intriguing, right? This is also a question then. Say, if we are assuming that just size based on size, if you are assuming that the size says that if size is normal, we'll consider it normal for the sake of discussion. Then why should we have it? Because if there are 1x IGF2 and IGF2R, both are present, normal. Both are not present, normal size, right? Then why have them in the first place, isn't it? Of course, there are many other functions that IGF2 are, but it is a good way of thinking and trying to feel puzzled by biology. And I would also encourage you to find, I already must have told you, try to find the evolutionary basis of genomic imprinting. Why is it there? Why did it evolve? Okay, the one way of saying it, if it is not there, what big loss is there? That's how we should think about it. And several researchers and scientists have their own opinions, and it's worthwhile to find their opinions and side with one or make your own opinion too. So yes, IGF2 has its own, this is IGF2R. They are all kind of integrated into the cell signaling network, and they do have several other functions. Okay, in different cells, they may have different functions. So it's just not about the size, but yeah, they may have functions, but it's worth thinking about it. As I already told, many of the imprinted genes are involved with in the growth or formation of placenta and the growth of embryo. Some things like IGF2 is here, and several other genes. So what we are learning is to have a proper understanding of, we are trying to understand all these mechanisms. Actually, what we are discussing is introduction of epigenetics. Epigenetics is just a part of one unit for us, but by itself is a huge course. And what we have tried to understand is the mechanisms of imprint, some examples, and also how they change over time period. As I said earlier, we want to change the cell from one form to the other form, one cell type to another cell type. That means we are wanting to change the programming of the cell. That is why we say it as reprogramming. We want to reprogram the new genetic reprogramming so that one cell becomes another type. And if you want to do that, all these mechanisms, the epigenetics and imprinting mechanisms, they all come into action. And that is why we are trying to understand epigenetics enough. So I hope we had good enough understanding about imprinting. It is complicated, and we will try to, over the next two classes too, we will try to have a better grip of it. I want to leave you with some points about some introduction to the next class, okay? So try to focus a little bit. We know about animal cloning. It is nothing but SCNT, or somatic cell nuclear transfer. We take a nucleus from somatic cell, we put it into an enucleated egg and see what happens. Animal cloning is done a long time ago, 1966 with frogs. The experiments were done, and Dolly is the name of the first sheep that is, or first mammal that is cloned. There are problems with cloning, and the problems first, of course, are epigenetic status, which will include both the imprinting as well as the genome program, that is the other epigenetic mechanisms. Which genes are silenced and which genes are active, expressed, and which genes are poised. There is also another issue is how to activate the zygotic genome activation. And one another problem that we discussed is with the telomere size. The size of the telomeres in the somatic cells are usually smaller, and therefore they may age fast, right? The organisms that have come out of animal cloning may age very fast. Yes. So here is, I'm going to give in this and next slides, I'm going to give you some introduction, which I already have discussed, but it will get a little complicated. Try to remember. What happens? We are just seeing it as DNA methylation only, okay? Forget about imprint and all this stuff at this point. So at this, what happens? We studied with imprints. We studied that. We learned that primordial germ cells, erasure happens, and then during gametogenesis, new imprint is established. In the sperm, everything happens much more faster because it's in one set. In mature oocyte, only upon fertilization, everything is completed. Cell division, meiosis. During the formation of, during spermatogenesis, there are a lot of things that happen. Around this stage, all methylation that is present, including that of the imprints, or DNA methylation is erased, that is demethylated. And during spermatogenesis, there is DNA methylation. There is histone methylation and histone deacetylation. All of these indicate that majority of the genes are repressed or silenced when in the sperm. All that the sperm has to do is to carry the nucleus into from the through uterus to the fallopian tube and meet the egg to fertilize it, right? So, and there is one more change that also happens because it has to be very tightly packaged. It is wound around protamines, and that is histones are replaced with protamines. In the sperm, the DNA is wound around protamines. You should probably remember that. And once the fertilization occurs, what will happen? This is the first. Here is HPF. This is hours post fertilization. This is another way of how the units are. This is if the IVF is it is done by in vitro fertilization, then you know when fertilization is done, and then you count hours from the time of fertilization. During that time, we have the first sorry, one second. Yes. So the male and the female. Second again. I needed the laser. Yes. This is the male pronucleus, and this is the female pronucleus, and cell division meiosis is being completed, and one of it will be expelled into polar body. So the DNA of sperm is completely methylated as it came from here, right? And now there will be rapid demethylation in this case. In the two of these, one of it still gets methylated. Remember that sperm has already finished the meiosis, whereas the egg is still undergoing meiosis, and only after the extrusion of the polar body is the meiosis of egg completed, right? So the methylation of egg, the genome of the egg is still continuing, whereas the sperm came with methylated DNA and it is getting demethylated here. So the male pronucleus the DNA of it gets majority of it demethylated, while the female pronucleus is still getting methylated. Later, these two will fuse and will form one nucleus. Okay. So here is the complex part, and this is the last slide. And I don't expect you to understand everything right away, but see if you can understand. Okay. So there are 4 colors depicted here. One of it is black, and one second. Yes, this is one. The red one is another. There is sky blue, and there is gray. There are 3 different ones represented. As we know, we have 2 types of genes, if you can categorize based on imprinting. Some of them are imprinted. That means the genes like IGF2 are imprinted, or H19, or IGF2 receptor, and so on. So some of them, if you take in, some of them are expressed from male, some of them are expressed from the maternal or paternal allele. The rest of the genes that are not involved with imprinting at all are called as the non-imprinted sequences or genes, which include all the housekeeping genes. They can also be cell-specific genes, spell-specific genes. They could also be all other types of genes. So in imprinted genes, there are 2 different. Black represents methylated, and gray represents non-methylated. And non-imprinted ones have 2, red and blue, maternal and paternal. This whole graph here is representing what happens to the whole genome from the time of primordial, before gametogenesis, until the whole body is grown. So if you see, this is trying to depict the methylation status of the genome, okay? And this is the time. So here, this is at the primordial germ cell stage. During that time, they might have come with whatever genome imprints and other methylation status that is happening. And during or around the formation of primordial germ cells stage, all the DNA is demethylated. All methylation is removed. Okay, so that's why it is showing as low. It doesn't matter if it is imprinted genes or non-imprinted genes, everything is demethylated. And during gametogenesis, this is formation of mature gametes, about that time, the whole genome is methylated. All of it is methylated except these genes, right? Here is the gray line. Except these genes, every other parts of the genome are imprinted or methylated. And upon fertilization, that is zygote formation, what we have seen in the previous, we have seen rapid demethylation of the male genome, right? So you might have the blue indicates paternal genome set, the male pronucleus, that gets rapidly demethylated. Whereas female, the maternal one, gets much more delayed demethylation. It is wonderful to really think that we all have been through these processes and we are the product of such kinds of processes, right? And demethylation is happening to every genes except these genes. What are those? They are methylated imprinted genes. Except these genes, other genes are getting demethylated. And once differentiation is happening, so some genes will get methylated, some genes will get demethylated, and epigenetic modifications will happen, all the others. The black and the gray do not change, right? They will remain the same for the rest of the lives. Okay, I mean, until in the early embryo stage. It doesn't mean the cells that we have now, right now, may not have the same imprinting status at this time. Not all the cells may be. I hope you understood a little bit. At least don't worry about understanding everything. It is all right. And this is an introduction to what I will discuss in the next class. And I hope we'll be able to understand it better. Anybody has any questions on this? Anyone, please? Okay, don't worry. We will discuss this again anyway in the next class.